I welcome you once again and invite you to look forward to a day of learning, sharing, and inspiration. So happy learning to all of you, and I would now like to invite our first speaker of the day, Dr. Jayant Hota. He is a senior consultant nephrologist in the Prastha Apollo Hospital, New Delhi. And his session is Introduction to Organ Transplant. So a little bit more about Dr. Hota. He's a gold medalist in MD with several national and international publications. He's actively involved in academics. He trains DNB students and organizes the annual GRASP meeting. And his expertise lies in kidney transplantation, glomerular disease, and electrolyte imbalances. So before giving my mic to Dr. Hota, I would just like to request all my faculty that please keep to the time. There would be a bell before the end of your session, which would be at 15 minutes, and then a bell at 2 minutes, and then it will be over. So also I will request our delegates that we have only time for 2 questions, not more than 2 questions. Otherwise, whenever you're having your tea and your lunch break, you are most welcome to interact with the faculty and clarify your doubts. So over to you, Dr. Hota. Thank you.
Thank you, ma'am, for the nice introduction. Good morning, sir, and good morning, everyone. At the outset, I want to thank Dr. P.N. Gupta, sir, for giving me this opportunity to deliver a lecture on introduction to organ transplantation. So as as you are all aware of organ transplantation, people are actively involved in, preparing patients for kidney transplantations. So without wasting much time, so we'll just briefly go into what is organ transplantation, and the whole throughout the day today you will be told about different intricacies, including the legalities of organ transplantation. So today, just in the introduction, what is organ transplantation, briefly we'll go through So this is the brief outlines. We'll be discussing in next maybe 10-15 minutes. What is organ transplantation? What are the different types of organ transplantations? Who are the potential donors? Would the little bit of immunology of transplantation, because you might be dealing with HLAs and DNA testing, and then little bit of the clinical organ transplantation, the rejections and anti-rejections therapy. So as you all know, organ or tissue transplantation is a surgical procedure in which a failing organ or tissues for that matter of a person is replaced with a functioning organ or tissue of another person, who again can be alive or dead, depending on whether you are doing a live transplant or a cadaveric transplant, or xenotransplantation where tissues or organs are taken from animals. And examples can be kidney transplantation, liver transplantation, heart, lung, intestine, cornea. Cornea is a tissue, it's not an organ, to be very frank. Bones, bone marrow. So these are different type of tissues and organ transplantation which are carried away. And it is a surgical procedure in which a healthy human being or maybe a dead one with healthy organs, we conduct a surgical procedures. If you go by the timeline, the first successful transplantation as far as the cornea transplantation concerned, it was first done in 1905. First successful kidney transplantation in 1950, first heart transplantation in 1967, first baby born of a transplanted ovary was in 2008, and the first successful entire face transplantation was done in 2013. Now coming to what are the different types of transplantation. So if you look at the different types of transplantation, it can be an autograft, it can be isograft, allograft, xenograft, a split transplant, or a domino transplant. So what is an autograft? Autograft is where the transfer of tissue organ from one own body to another side. Like for example, a skin grafting. You are taking from a, from a concealed part to, let us say, a face or somewhere. That is called an autograft. Isograft is when the transfer of tissue organ between genetically identical individuals, like for example, identical twin giving one kidney or liver or something to his or her twin. But allograft is the one what we keep on doing. Allograft is what? Allograft is an transfer of organ or tissue between genetically different individuals of the same species. Like for example, a human being giving organ to another human being. Okay, that is called allograft. Whereas if an animal is giving, if you are taking tissues or organ from an animal, that's called a genograft. Genograft is the transplant of organ or tissue from one particular species, like for example, pigs and other species, to human being. That is called genograft. There can be split transplant. Split transplant, that is a process in which an organ of a deceased donor, especially liver, because liver is can be split into two parts, and one part is given to one individual and another part is given to another individual. That can be a split transplant. Or there can be a domino transplant, in which the complete set of organs is transplanted, like for example, a cystic fibrosis in which the lungs is replaced. That's called a domino transplant. Now this looks like a busy slide, but this is important. In the sense, when we are transplanting, what is happening is like we are putting one foreign tissue, which is not ours, into our own body. So what happens is like the body tries to defend it. So what are the two defenses? We have one inborn defense, that is the innate or inborn non-specific defense, and one acquired defense. So innate defense can be skin, like one of the first layer of defense is skin, or there can be some internal second line defense, like for example our neutrophils and phagocytes and all those things. That we are not much concerned. What we are concerned is about the acquired defense. And that can again be divided into active immunity and passive immunity. In active immunity, what happens is the T cells and the B cells, they get activated. Whenever one foreign tissue is put into our body, T cells and B cells will always try to defend that. Will try to kind of remove that organ. How do they do that? By doing a rejection. That is what we are more concerned about. That is why there is a lot of this HLA testing and all those things. And something called passive immunity, in which what we do is we give antibodies to treat that rejection or something. Sometimes we use IVIGs and other things while treating rejections. Now, who can be a source of kidneys for a kidney transplantation? So, kidney donors can be either a living donor, we are concerned about living donation today, or it can be a deceased donor, in which the patient is dead. In the living donor, again, it can be related, okay, or they can be living but unrelated donor. And third, these days, it has become very popular these days, is altruistic donor. In altruistic donor, what happens is, let us say my family member is there. The family member is donor, he or she is a donor. But because of some reason, because of the high DSAs or because of some incompatibility, the patient is not able to donate to me. So what will happen is the donor will donate to some other person, that person will donate to me. So that is a kind of non-directed donation. So these days, that also people are doing. That is called altruistic donor. And deceased donor can be divided into, donation after brain death. That is the most common one. That is called DBD. Or these days, we are also doing donation after circulatory death, in which the heart has stopped pumping. Still, you can remove the, organs. If you can do it, that again can be divided into controlled and uncontrolled. Let us say somebody is brain dead. Okay. So many times, what happens is like the family will not accept brain donation, brain death donation. They will say, oh, heart is pumping, how are you saying that the patient is dead? Okay, so what happens is like you can controlled way, you can stop the heart beating. Okay, so once the heartbeat stops, you can remove the organ. And there's one uncontrolled way. Let us say, somebody develops a, heart attack. Somebody is in the ward or somewhere, the patient develops heart attack and the patient has died. Okay, there is no heart, no brain, no activity. That is an uncontrolled way of, taking an organ. Because in that case, what will happen is like all the organs will be badly damaged because of the ischemia. Because the heart is not able to pump, blood circulation will be hampered, so it will be badly damaged. Whereas in controlled donation, what happens is like you are only stopping the heart. So at what time you are going to remove the organs. So controlled, donation after circulatory death is better than uncontrolled one. Now this everybody knows. Okay, so recipient of O group can only take, donation from the type O. A will take from O and A groups, B can take from O and B, and AB can take from every other group, like O, A, B, AB, all four can do that. These days we are able to do the ABO incompatible transplant. So this is when there is ABO compatibility. But these days we are also able to do ABO incompatible transplant in which any blood group can donate, organ to any other blood group. So that is a separate thing. Now what is HLA typing? Most of these, before transplantation you guys are doing HLA typing. So what is HLA typing? In this test what happens is that the donor's antigens expressed on the surface of leukocytes or the genes that are matched with that of the recipient. So what happens is, there are two types of HLA, again HLA1 and HLA2. HLA1 is present in all nucleated cells except RBCs. It is there in RBCs also. So there is one particular antigen which makes us specific. So who I am, depending on my HLA antigen. We may be looking same because we are all human beings, but we are different. How are you different? Different because we have got different set of HLA antigens, different set of genes and antigens. So what happens is when you are trying to correlate whether somebody is a father or somebody is a sister, somebody is a brother, just by looking at the HLA, we can find out whether the patient, the particular man or woman is related to the other man or woman. The closer the HLA antigens on the transplanted organ match the recipient, the more likely that the recipient body will not reject the transplant. So not only by doing HLA, not only do we establish the relations, but if you are getting a good HLA match, then probably the success of kidney transplantation will be better because they are genetically matched. So the value of HLA matching between donor and recipient varies in different solid organ in transplantation. So we in kidney transplantation these days, we are not much worried about the HLA mismatch. Okay? Even if somebody is like a dead body is donating, or let us say a wife or husband is donating to his or her spouse, there the HLA antigen matching won't be there because they are not related. But we are not concerned. In kidney transplantation, we are not much concerned. But there are certain instances in which HLA matching is very important. In kidney transplant, there is substantial benefit if all the polymorphic HLA alleles are matched. But if you can get a good HLA match, then the success of kidney transplantation will be better. So this is what I was telling. This is the chromosome, and this is where the short arm of chromosome number 6 it is represented. And they are again HLA class 1, 2, and 3. HLA1 is ABC and 2 is DPQR. So this is how they look. HLA1 and HLA2, they are present on the surface of the leukocytes. Their chromosome is there in the nucleus, but the antigens are present on the surface of the leukocytes, and they are very important. So this is what it is. If somebody has got a, like you can see here, this one and this one, let us say this is mother, and this one, this is father. So the children will have one copy of this, one copy of this, and one copy of this. So you can look at this, the yellow from father, it is there, yellow from father, and the purple from mother. They have come to the children, like that. So one copy from father and one copy from mother. So if there are two for mother, two for father, one will come to each, okay? So that is how we can make sure whether the patient, the donor is a child, or, whether the mother, let us say you want to, establish a relation between mother and child, you can easily make out by looking at the HLA antigens. Now, sometimes what happens is like there is no HLA matching. Let us say somebody has come, somebody will say, oh, he's my cousin or something. There it is very difficult to find out a HLA match. So what we do is we go for a DNA match. But the problem with DNA match is that even we as humans will resemble with primates, like for example, monkeys and gorillas and chimpanzees, to the extent of more than 90%. If you look at a primate genetic structure and a human genetic structure, the matching can be as high as 90% because we have all come from the same ancestry. That is the problem with DNA. So in DNA, it is very, very difficult to establish whether you have, and this is a very busy slide, what you can see. If you are, doing a DNA match with your father, that there can be 50% chance that you will get a match. But 50% again chance is that you will not find a match. And it goes down. If you go to the, second generations, uncle, aunt, it is 25%. Go to the next cousins, it will be 6.25%. The second cousin, it will be 1.5%. So getting a match and just looking at the DNA match and saying that they are related is very, very difficult, unless you have got all the genetic structures. So most of the labs, what they do is that they look at only 16 to 18, genes, and they compare whether they are related or not. So if, let us say you have taken 18 genes and more than 50% are, matching, then probably they have some relation. But it is very difficult to say whether they are related or not. You can say, yes, there can be some relation. That is the problem with DNA matching. And then this is, before transplantation, we do a cross match, whether the patient is, whether we can donate, a particular donor can donate organ to another recipient. by looking at the CTC cross match and the flow cross match. One is cell based, one is solid based. These days we are doing the Luminex also. The Luminex will tell us whether the patient has got donor specific antibodies, and accordingly you have to be prepared, because otherwise there will be rejections. And what are the different types of rejections? Rejections can be hyperacute, accelerated, acute rejection, chronic. These days we are not getting hyperacute rejections because of the good cross matching. But accelerated acute rejections are always there, and that can be in the few days, few hours to few days, or acute rejections can be 1 week to few months. Or there can be chronic rejections after 3 months. So this is how, the rejections takes place. The antigen presenting cells will present this antigen to the T cells, and then the T cells will start activating other T cells and B cells, and they can produce antibodies and they can kill the endothelium, and that is how the rejections takes place. So in rejections, we can have activation of the complement that will damage the endothelium. There can be interaction of the HLA antibodies with the endothelium, and there can be antibody dependent cytotoxicity that can damage the organ, and the patient will have a rejection, full-blown rejections. So what are the 3 major categories of preventing rejection? So it can be pre-transplant, peri-transplant, and post-transplant. In the pre-transplant, by doing a good HLA matching, you will find out, yes, whether the patient is matching HLA, and the chances of rejections will be less. If there is some kind of a, donor-specific antibodies, you can go for a desensitization. In the peri-transplant period, you have to decrease the cold ischemia time that is done in the, operation theater, and you have to enhance the perfusion and individualize induction in peri-transplant period. Post-transplant period, if the patient has developed, rejections, you have to treat the rejections by using different anti-rejection therapy. So this is how we treat hyperacute rejections. These days are very uncommon. Once there is hyperacute rejections, you have to remove the kidney because that kidney is not going to work. But if it is an acute rejection, there are medications to treat the acute rejections. So coming to the living donor kidney transplantation process, like let us say in a hospital, if you are doing living donor kidney transplantation, what is the chance that it will be a successful one? And that requires a comprehensive package, and that requires not only a centralized structure, a mandate of timely intervention, an equitable funding model, commitment to collaboration, cultivating the distributed expertise, financial burden on the donors and recipients. That has to be, there are some, at some places the government has to intervene to give some subsidy to the patients, and there are certain places in which we can do by increasing our, the quality of care and quality of surgery. So it's a holistic way of, looking at kidney transplantation, or for that matter, any other transplantation. That requires some government help from the hospital side, from the manpower, from the surgeons, from you guys who are behind any successful organ transplantations. So it's a holistic one. Only one thing can't make sure that the transplantation will be good. Let us say surgeons are good, but other things are not working properly, then the transplantation will not success. You guys are good, but surgeons are not working, or the nephrology is not working properly, then again the successful transplantation won't be there. Okay, so I'll conclude with that. Thank you. If there is any question or anything, I'll be happy to answer.
Thank you, Dr. Hota, and special thank you for keeping to the time.